Hello, today we'll talk on the topic RNA interference. What is the RNA interference? It is the gene silencing process. Why? Why it is important? Because it is the cellular defense mechanism protect the cells against the viruses. How? How the transgenic plant is produced by using the RNA interference technology? Let's understand this concept in detail with some example. For example, you have the big tobacco plant. Suddenly the tobacco plant production will decrease. The farmer upset. What will happen if there is no tobacco plant? Here, the most of the nematode that is called as Meladovine incognita attack onto the roots of the tobacco plant, produce the root knot disease. They absorb all the essential nutrients, so the plant will not get any nutrient. Ultimately, the plant will die. Very serious condition. We have to protect the plant. So we use the RNA interference strategy. In this strategy, what we use? We introduce the gene. Gene means exactly it is the nematode specific gene. The gene which is obtained from the nematode itself. This is responsible for the survival of the nematode, and this is inserted into agrobacterium vector. When it is inserted, this complex is ready for insertion into the tobacco plant. When it is inserted into the tobacco plant, what will happen? It will undergo the transcription process. Normally, the genes undergo the transcription process. Similarly, it undergoes the transcription process. In case of transcription, it forms the two strands of RNA. One is the sense strand, another is the antisense strand. Both strands are complementary to each other. When they are joined together, they form the long double-stranded RNA. Now, this long double-stranded RNA is very long, so it is broken down into the small pieces to form the siRNA, that is the small interfering RNA. Now, this small interfering RNA will unbind and they bind with a protein molecule to form the siRNA protein complex. Now this siRNA protein complex is ready for attacking. Now next part of the story we'll see what happened in nematode. In nematode there is a DNA. The DNA which undergo the transcription to form mRNA. mRNA undergo the translation to form protein molecule. The proteins are nothing but the enzymes which are responsible for the survival of the nematode. When the nematode engulfs the tobacco plant, what will happen? This siRNA protein complex attack on mRNA, will block the mRNA, silence the mRNA, and will not allow the continue the chain. And so the translation process is inhibited over here. Once the translation process inhibited over here, there will be no protein synthesis. No protein synthesis means no enzyme, and ultimately the nematode will die. So by this process we can protect our tobacco plant. Hope you understand this concept very well. Thank you very much.